All right, engineers, in this video, we're going to continue off with the glycogen metabolism, but we're going to end off in this video talking about the regulation of glycogen metabolism. Because understanding the pathway is significant, that's important, but specifically understanding what's controlling whether it's getting broken down or being synthesized is even more significant. Okay, so let's say I take the two main enzymes that are being regulated in that glycogen metabolism process. So let's say that I take this enzyme here, and we're going to say that this enzyme was the glycogen synthase. So glycogen synthase, you'll remember he was the one that was forming those alpha-1,4 glycosidic bonds, elongating the actual glycogen polymer. The other one that we're going to talk a lot about, look at this bearded dude, is going to be what's called glycogen phosphorylase. If you'll remember, this was the guy that was reaching into a satchel, pulling out those phosphates and adding those phosphates into the actual glycogen to form glucose 1-phosphate. These two enzymes are the main enzymes that are being regulated heavily in the glycogen metabolism video. So if we were to kind of just make like just a simple kind of concept here, technically, let's say that I have glycogen here, and I have glucose. Let's do glucose in a different color. Let's put glucose in red. Glucose. If I'm going from glycogen to glucose, that's called glycogenolysis. So if I'm going this way, if I'm going from glycogen to glucose, this process is called glycogenolysis. But then, if I'm going from glucose to glycogen, this is called glycogenesis. So again, what is this air? If I were to kind of write on this side here, this side here is going to be glycogenesis. Right? Glycogenesis is going from glucose to glycogen. And then over on this side, you're going to have glycogenolysis. And this is going to be taking the glycogen and breaking it down to glucose. If you'll remember, this enzyme, the glycogen phosphorylase, is controlling this step. He's one of the main enzymes involved in this step, breaking the glycogen down into glucose. Whereas this enzyme, the glycogen synthase, he was involved in the building up of glycogen. He was responsible for being able to synthesize glycogen. Now, it's not enough just to know that. We have to know what's controlling this enzyme and what's controlling this enzyme. It's not too bad if we think about it logically. Glycogen synthase wants to make glycogen. So, what would be a stimulus of this reaction? The main powerful stimulus, allosteric, because we're going to do two types of regulation here. So there's going to be two types of regulation. One type of regulation is going to be with respect to allosteric regulation. Allosteric regulation. In other words, meaning that this molecule binds to some other site other than the active site, and it changes the overall three-dimensional structure of that molecule, whether it either activates it or deactivates it. It depends on the molecule. The other type of regulation that we're going to focus on is hormonal, and that will be the next one that we'll talk about after allosteric. So hormonal. How are hormones influencing these enzymes? Okay, now, glycogen synthase, let's think about this logically again. Let's say that I have a lot of glucose 6-phosphate. What would a lot of glucose 6-phosphate indicate? Okay, that would indicate that I have high blood glucose levels, because if there's a lot of glucose 6-phosphate, that means that a lot of glucose is getting brought into our cells and getting phosphorylated. That means that the blood glucose levels are high. If the blood glucose levels are high, what hormone is getting activated? Insulin. And insulin loves to stimulate glycogenesis. So wouldn't this be a stimulator of this enzyme because he wants to synthesize glycogen? Yes. So this molecule will bind onto some allosteric site and stimulate this molecule. But think about it logically. Let's come over here to this guy, this glycogen phosphorylase. Think about this now. What if I have really, really high glucose 6-phosphate levels? If it's stimulating in that enzyme, we wouldn't want this enzyme to be stimulated at the same time. So wouldn't we want this same stimulus to inhibit this enzyme? Yes. So this is going to inhibit this enzyme from functioning. That is one of the most powerful allosteric stimuli of the glycogen synthase. Okay? And that is going to be what we're going to talk about mainly here. Now, that's going to be the allosteric stimulator. Now let's talk about something that's going to inhibit this process, but not specifically inhibit this enzyme. We're going to talk about what's inhibiting this enzyme or what's stimulating this enzyme. Okay, so we know glucose 6-phosphate inhibits this enzyme. What else could inhibit this enzyme? You know, if you have a lot of glucose 6-phosphate, you're making a lot of, specifically a lot of ATP. And if you're making a lot of ATP, that means that there's a lot of Krebs cycle activity, a lot of glucose being broken down. So in that case, if we're already utilizing glucose through glycolysis and the Krebs cycle and the electron transport chain, we don't really need glycogen to be broken down to glucose because it's just going to make us make more ATP. And if we have too much ATP, that's going to stop this process. How? Significantly large amounts of ATP is going to inhibit this enzyme. And if high amounts of ATP inhibit this enzyme, he can no longer break glycogen down into glucose. Why? Is that a good thing? Because if he keeps breaking glycogen down into glucose, there'll be too much glucose, too much glycolysis, too much Krebs cycle, and then too much ATP. And too much ATP, our cell can only store so much ATP in the cells. So ATP will inhibit this enzyme. Another thing, free glucose. But we have to be very careful when I mention this one. So high amounts of free glucose can also inhibit this enzyme. And it should make sense because if there's a lot of glucose, that means that there's going to be a lot of glucose coming into the cell. The blood glucose levels are high. If our blood glucose levels are high, we're not going to want glycogen to break it down into glucose and make them even higher. That's going to make it worse. So we want to inhibit this guy. But this is only happening in the liver. Glucose is only inhibiting this glycogen phosphorylase in the liver. In the muscles, this is not inhibiting it, okay? So we need to make sure that we're very careful with that. Glucose is only inhibiting glycogen phosphorylase in the liver, not in the muscles. Okay, now we need to do something else. Now, there's a weird component to this glycogen phosphorylase. There's something else that we can do to this guy. Okay. Oh, before we do that, one other thing. Before we do this one, there's another stimulus. There's a stimuli to this. If you have high amounts of ATP, right, that means that you have low amounts of ADP. Okay, so remember, if you guys remember this concept, high amounts of ATP means low amounts of ADP, right? Because what's happening is we're forming a lot of ATP, so not a lot of ATP is getting broken down into adenosine diphosphate and an inorganic phosphate. But if your ATP levels are low, a lot of this ATP can be getting broken down into a lot of ADP in an inorganic phosphate. So what if our ATP levels are low? If our ATP levels are really, really low, what is that significant about that? That means that we're not having a lot of glycolysis. We might not have enough glucose in the bloodstream. There might not be enough Krebs cycle activity, not enough activity in the electron transport chain. We need to make more ATP. Okay, well, this enzyme should be stimulated, break down glycogen to glucose, get that glucose into glycolysis, Krebs cycle, and make more ATP. How does it do that? Now, it's not ADP that does this. It's something very, very special that does this. It's another breakdown product. Because you know ADP, you can actually take ATP, and ATP in your muscles can actually get broken down even further than ADP. It can get broken down into AMP and pyrophosphate, which just means you're having, pyrophosphate is just two inorganic phosphates linked together. Now, if I have this AMP being formed because of excessive ATP breakdown in the muscles, this will stimulate this guy and let him know to start becoming active. So if there's high amounts of AMP, that will stimulate this enzyme. But this is only coming from the muscles. This is only coming from the muscles. It's a breakdown product whenever we have that cross bridge formation. And whenever the muscles contract, they actually build up a lot of AMP, and AMP can stimulate this enzyme to break down more glycogen into glucose so that glucose can undergo that cellular respiration process to make more ATP for muscle contraction. Okay, now again, that's only happening in the muscles. Now, there's another weird thing about this. There's an enzyme that controls this enzyme's activity. Okay, so let's come down here. Now, we have an enzyme. This enzyme is called Phosphorylase Kinase. But there's two forms of this enzyme. One form is inactive, and this inactive form is type B. So phosphorylase kinase B is the inactive form of this enzyme. But whenever he is active, he can get converted into, what do you think, guys? Phosphorylase kinase A. And A is the active form of this enzyme. So whenever it's in phosphorylase kinase B, it's actually going to be inactive. But whenever it's in the form of phosphorylase kinase A, it'll be active. What triggers that process to occur? So what triggers this conversion of phosphorylase kinase B to phosphorylase kinase A? And why is that relevant to glycogen phosphorylase? Okay. Now we're going to get into another process. Now we have to get into a little bit of the hormones. So we'll come back to this and see how this is affecting it. So inside, on your liver cells, you know you have these things called G protein coupled receptors. So these G protein coupled receptors are going to be special because they're responding to a specific types of molecules. What are these molecules that it's responding to? Remember I told you that glycogenolysis is primarily stimulated by glucagon and norepinephrine and epinephrine. I told you that we would come back to that. Now it's time. So now who are the molecules that can bind here? So what is this molecule? What could it be? It could be a plethora of molecules. What molecules? This could be glucagon. This could be epinephrine. This could be norepinephrine. Any of these molecules. And it's binding onto these receptors, these G protein coupled receptors. When it binds onto this G protein coupled receptor, you know you can activate G stimulatory protein. And you know G stimulatory protein can then do what? If you have G stimulatory protein, he can bind to GTP, which makes him active. When he becomes active, what can he do? He can then go and stimulate a effector enzyme. So there can be some type of effector enzyme embedded into the cell membrane. Let's say here is our effector enzyme. You guys already know this enzyme. This is called adenylate cyclase. What does this enzyme do? It takes ATP and converts it into cyclic AMP. And then cyclic AMP activates protein kinase A. Okay. So now we'll come back to this protein kinase A in a second. Okay. This protein kinase A is going to be occurring whenever what? Whenever glucagon, epinephrine, and norepinephrine are being released. When would these hormones be released? They would be released whenever our blood glucose levels are low. If our blood glucose levels are low, we're going to want to stimulate glycogen breakdown into glucose, specifically called glycogenolysis. Now, how does that happen? You see this protein kinase A? He's going to come over here and he's going to phosphorylate this kinase. So look at what happens here. Here's our protein kinase A. This protein kinase A can come over here and it can phosphorylate this molecule. So now look, he's going to phosphorylate him. If he phosphorylates this phosphorylase kinase B, it activates this enzyme. When this enzyme is activated, then what is he going to be? Phosphorylase kinase A. So the phosphorylated version of this enzyme is active. So now he is active. Okay? And again, who is doing this? Specifically, protein kinase A. So protein kinase A, which is coming from what? Coming from glucagon stimulus, or norepinephrine stimulus, or from epinephrine stimulus, is going to activate phosphorylase kinase B and turn him into phosphorylase kinase A. Now what is going to happen? Phosphorylase kinase A is going to kick this guy into gear. He's going to come over here and he's going to put phosphates right on this guy. Look, he's putting phosphates on his armpits, right? When he puts phosphates onto this actual enzyme, he's going to become activated. Okay, when he becomes activated, he's then going to do what? Stimulate this step, converting glycogen back into glucose. So now that should happen. But again, what is happening? Specifically, glucagon, epinephrine, norepinephrine are being released when our blood glucose levels are really low or when we're in a stressful situation. Binding onto G protein coupled receptors, activating the G stimulatory, binding with GTP to become active, activating adenylate cyclase, adenylate cyclase converting ATP into cyclic AMP, cyclic AMP activating protein kinase A, and then protein kinase A is doing what? It's phosphorylating phosphorylase kinase B, turning him into phosphorylase kinase A. And then phosphorylase kinase A phosphorylates glycogen phosphorylase, which stimulates this enzyme, who then converts glycogen into glucose. There's one more mechanism that can do this also. You know, glucagon, epinephrine, norepinephrine, they have different types of adrenergic receptors, like alpha adrenergic receptors and beta adrenergic receptors. Let's say over here, I have another receptor, specifically, another type of receptor. And this receptor is another G protein coupled receptor. But whenever epinephrine and norepinephrine bind onto this G protein coupled receptor, it activates a different type of G protein. Let's say this specific G protein is going to be GQ. What GQ does is it's still going to bind onto GTP and become active. When he becomes active, he goes and activates a different enzyme. So let's say here's this different enzyme. This enzyme's special because it's a phosphodiesterase enzyme. This enzyme is called Phospholipase C. And what happens is when this GQ stimulates him, Phospholipase C does something really cool. He takes and breaks down a molecule which is called PIP2. He breaks him, and he breaks him into two components. One is called diacylglycerol. The other one is called IP3. You know that there's certain types of storages that have what's called the endoplasmic reticulum in these cells? And in these endoplasmic reticulums, they're rich in calcium. You know what it's going to do? It's going to pump out calcium ions. This calcium is then going to bind to a protein called calmodulin. Calmodulin. And when calcium binds to the calmodulin, this can stimulate the conversion of phosphorylase kinase B to phosphorylase kinase A. So again, let's review this one more time. Epinephrine and norepinephrine can also bind onto different types of G protein coupled receptors on the liver, right? And when it does that, it activates GQ. GQ activates phospholipase C, which cleaves phosphoinositol diphosphate into diacylglycerol and inositol triphosphate. Inositol triphosphate binds onto receptors on the endoplasmic reticulum and pushes out calcium. Calcium binds onto a molecule called calmodulin. And then that calcium-calmodulin complex will stimulate the conversion of phosphorylase kinase B to phosphorylase kinase A. If phosphorylase kinase A is activated, he'll phosphorylate glycogen phosphorylase. If glycogen phosphorylase is activated, he'll convert glycogen into glucose. Holy crap, that was a lot, right? But it should make sense. But notice protein kinase A, he's not selective. He doesn't just say, I'm going to phosphorylate this guy and not this guy. He also phosphorylates this guy. This is our glycogen synthase. So you know that protein kinase A, look what can happen. You know glycogen synthase actually exists in two forms also? He can exist in two forms. It's the same thing as the phosphorylase kinase B. So let's say I have here glycogen synthase. But specifically, this is the B type. And then over here, I have another one. And this is called glycogen synthase, and this is type A. If I have that protein kinase A come into play. So let's say here's my protein kinase A. If that protein kinase A phosphorylates this this glycogen synthase that's in the B form and converts it into the A form. Sorry, this is not going to happen. It should actually be the opposite. If this glycogen synthase is in the active form and he gets phosphorylated, by protein kinase A, when this protein kinase A phosphorylates the the glycogen synthase protein, it converts him into the inactive form. So this is the active form, and this is the inactive form. So again, what is protein kinase A doing? Protein kinase A is adding phosphates onto glycogen synthase. When he adds the phosphates onto glycogen synthase, which is in the active form, he'll then be converted into the inactive form. So that should make sense. And what would that do to this enzyme? He'll then be inhibited. And if protein kinase A phosphorylates glycogen synthase, which converts him from the active to the inactive form, can he take glucose and convert it into glycogen? No. And so this process will be inhibited. So we can say in a weird way that protein kinase A is inhibiting this enzyme because he's doing what? He's putting phosphates on him. And putting phosphates on this enzyme inhibits this enzyme. Now we come to the hero who's going to save them all. This is called insulin. So let's say over here I have another receptor. Let's say here's another receptor. And this receptor is specifically for insulin. So here's my receptor. And this is different. This is a tyrosine kinase receptor, right? And we're not going to go over the pathway in this, but we're just going to say specifically what's happening here is that insulin is going to be released whenever your blood glucose levels are high. So whenever you have high blood glucose levels, insulin's going to be released. Insulin's going to activate this receptor. When he activates this receptor leads to the activation of very specific proteins, and these proteins are called phosphoprotein phosphatases. So who's activating these proteins, these phosphoprotein phosphatases? This is going to be the presence of insulin. So insulin is stimulating the formation of these phosphoprotein phosphatases. Okay, now these phosphoprotein phosphatases, when they're formed due to insulin, due to high blood glucose levels, these phosphoprotein phosphatases are going to do something special. So you see these phosphates that were put on by specifically the protein kinase A because of glucagon and norepinephrine and epinephrine. Well, look what happens here. I can do this exact opposite reaction. I can take glycogen synthase from the B form, which is the inactive form, and put him back into the active form. How can I do that? With these phosphoprotein phosphatases. I'm just going to denote them PPP, right? So phosphoprotein phosphatases. These phosphoprotein phosphatases are going to do what? They're going to remove phosphates from the glycogen synthase, which is in the inactive form, the B form. So now look, these phosphates are going to be removed from the glycogen synthase. If the phosphates are removed from the glycogen synthase, now the glycogen synthase goes from the inactive form to the active form. Who is doing that? Specifically these phosphoprotein phosphatases, which are coming from insulin. So again, one more time. Insulin is released whenever there's high blood glucose levels. He's stimulating these specifically these tyrosine kinase receptors, activating phosphoprotein phosphatases. Phosphoprotein phosphatases will rip the phosphates off of this glycogen synthase. So you can actually show it like this. Look, he's going to come into play, and what is he going to release off? What will he pull off of this glycogen synthase? He'll pull off phosphates. When he pulls off the phosphates, that stimulates this enzyme. So again, how is it happening? Let's say that this glycogen synthase is in the inactive form due to protein kinase A. Then what happens is we can take it from the inactive form to the active form, type A, whenever we have phosphoprotein phosphatases ripping those phosphates off of the inactive glycogen synthase, turning them into the active form. If this happens, then what happens to this enzyme? He is activated. And if he is activated, he can take glucose and convert it into glycogen. Last but not least, these phosphoprotein phosphatases are not specific also. So you know what they can do? You know how we were adding phosphates onto this phosphorylase kinase B to convert them into phosphorylase kinase A? These phosphoprotein phosphatases can also come over here. And look what they can do. They can remove what? They'll remove the phosphates off of this phosphorylase kinase A. When they remove these phosphates off of the phosphorylase kinase A, then what will happen? This then will convert back into the inactive form. So again, what happens? Phosphoprotein phosphatases act on the phosphorylase kinase A, rip off the phosphates, and he'll go from the active form back to the inactive form. But he's not done. See these phosphates that this guy put onto this guy's armpit? These phosphoprotein phosphatases are going to come over here. And what are they going to rip off of this glycogen phosphorylase? They're going to rip off phosphates. When they rip off the phosphates, then what's going to happen? This glycogen phosphorylase is no longer going to have phosphates on him. So will he be inhibited or will he be stimulated? He will now be inhibited because he was activated by phosphate groups. When you pull those phosphate groups off of him, he is then inhibited. So if you rip these phosphate groups off of the glycogen phosphorylase via the activity of these phosphoprotein phosphatases, it'll inhibit this enzyme. If this enzyme is inhibited, he can no longer break down glycogen into glucose. So that's why insulin stimulates glycogenesis, and epinephrine, norepinephrine, glucagon stimulate glycogenolysis. If you phosphorylate this enzyme, he's activated. If you dephosphorylate him, he is inactivated. If you phosphorylate this enzyme, he's inactivated. If you dephosphorylate this enzyme, he's active. Okay, now, last thing I want to tell you guys out of this whole video is that, you know, glycogen phosphorylase and the debranching enzyme are not the only enzymes that are involved in that glycogenolysis pathway. There's one other enzyme that I want to mention with you because I'm going to tell you guys what can happen when some of these enzymes are dysfunctional. There's one other enzyme that can account for about 1 to 2% of glycogenolysis. Most of them is going to be, if you guys remember from the glycogenolysis video, it was primarily just the glycogen phosphorylase and the debranching enzyme. 1 to 2% can be done by specifically lysosomes. In these lysosomes, we have what's called this acid. We can actually call it acid maltase or alpha-1,4-glucosidase. So acid maltase or alpha-1,4-glucosidase. Why am I telling you this? Because there is a disease that can occur whenever these lysosomes are having a deficiency or a mutation within this acid maltase or this alpha-1,4-glucosidase. Whenever these people are deficient in this actual enzyme, it can cause significant types of problems. Primarily, it can cause a lot of damage to their heart. And if that is occurring, they call this disease, very, very dangerous disease, they call it Pompei's disease. And this is specifically a glycogen storage disease type 2. Okay? Now, conversely, you know how you have this enzyme, this glycogen phosphorylase enzyme? He's extremely important also. If this glycogen phosphorylase enzyme is deficient, he can also have a mutation in this guy too. And here's the way I like to remember these types of, I kind of go down in like a weird order. So I go like this. Okay, so I go down like this. So I kind of remember these guys in a specific way. So I'm going to have a different type of glycogen storage disease for each one of these guys. So this is going to be my glycogen storage diseases. I'm just going to give you a couple of them, the more significant ones. One of them is actually going to be von Gerke's disease. And von Gerke's disease is actually type 1 glycogen storage disease. And what happens is, in this glycogen storage disease type 1, you're lacking a specific enzyme. That enzyme is called glucose 6-phosphatase. Then, we already know this one. If you have P, this stands for Pompei's disease. And in Pompei's disease, you're lacking a acid maltase enzyme. Then, if you go down again to C, C is standing for Cory's disease. Okay, this is for Cory's disease. And then you can keep going down. Okay, so let's keep going down. So, Anderson's disease. And then you can also have another one, which is going to be McArdle's disease. And the last one is actually called Her's disease. So here's how I like to remember how you can remember what enzymes are dysfunctional. So you see this C, what comes after C? D. And you know what actual enzyme is actually going to start with D? This is debranching enzyme. If there is some type of deficiency in the debranching enzyme, this can cause Cory's disease. What comes after A? B. This is the deficiency in branching enzyme. So if you have a deficiency in the branching enzyme, this can also cause a glycogen storage disease. Then, the M in McArdle's disease, if you kind of look at the M there, that M in the McArdle's disease, this is the deficiency in the glycogen phosphorylase, but specifically in the muscles, because they behave differently. So this is glycogen, I'm going to put GP, glycogen phosphorylase, but specifically in the muscles. Then if you see Her's disease, there's an H. You notice the glycogen phosphorylase enzyme that's also deficient in this one? But the glycogen phosphorylase, what does H stand for? Hepatic, liver. This is the liver glycogen phosphorylase. So here's how I like to remember these guys. Now that we have all this, I can go down in an order. This is glycogen storage disease type 1. This is glycogen storage disease type 2, type 3, type 4, type 5, and type 6. So if you have glycogen storage diseases, any deficiency in these enzymes that we've mentioned throughout all of these series of videos can cause significant damage on the heart, on the liver, on even the spleen. Different types of tissues can be extremely damaged due to these glycogen storage diseases. If you wanted to remember any of these, the most significant, the most damaging, the most dangerous one is von Gerke's disease. Usually these children never live past the age of 1 year old. So it's an extremely dangerous disease. So again, von Gerke's is the deficiency in glucose 6-phosphatase. Pompe's is the deficiency in acid maltase. Cory's disease is, remember, C, what comes after that? D, debranching enzyme. Anderson's is what comes after A, B, branching enzyme. McArdle's, remember the M is for muscle glycogen phosphorylase. And then H for Her's disease, remember that's hepatic or liver, the liver glycogen phosphorylase. Any deficiency in these enzymes can cause significant catastrophic effects on the body. All right, engineers, in this video we covered a lot about the regulation of glycogen metabolism and what can happen whenever these breakdown processes in this glycogen metabolism occur. I hope it all made sense. I hope you guys enjoyed it. If you guys did, hit that like button, subscribe, please, and leave some comments down in the comment section. We look forward to hearing from you guys. All right, engineers, until next time.